Hello, welcome to your own channel, Curious Corners in Biology, where we discuss and delve into the curiosity aspect in biology, where we think about hows, if, whens of this subject. Okay, to make it more interesting, as simple as that. Okay, so today we are coming up with our series of questions. The first question which comes to our mind is about Golgi complex. Usually in eukaryotic cells, Golgi complex is multicisternal. That means many cisternae are present in Golgi complex. But in fungal cells, in certain fungal cells, Golgi complex is unicisternal. Why so? So obviously we know Golgi complex is a cell organelle found in eukaryotic cell involved in lipid and protein modification. Okay. And it is also involved in packaging biomolecules in the cell to be utilized for various functions as well as to be transported outside the cell or secreted outside the cell. Now, the case with certain fungal cells is that they will have unicisternal Golgi complex. That means the cisternae, the flattened membranous sac, is singular in the hyphal cell. Now, the only reason for this which has been found is that such Golgi complex would help in polarized hyphal growth of the fungi. What do we mean by polarized hyphal growth? This is hyphal growth in one direction only. This is due to, of course, accumulation of modified proteins and lipids at the tip of the hyphal cells, which will drive polarized growth of the hyphal cells. So far only this reason is connected with the presence of unicisternal Golgi complex in fungal cells. Okay. The second question is quite common, that why do we consider ATP as the energy currency of the cell? So the full form of ATP, as we know, is adenosine triphosphate. This biomolecule is made up of three components: nitrogenous base, that is adenine; ribose sugar, which is a pentose sugar, 5 carbon containing sugar; and phosphate groups. There are three phosphate groups which are negatively charged and covalently bonded. You can say them as three phosphates as well. Now, usually when the molecule is intact, then the phosphates are together. But when ATP is broken down, which is called as hydrolysis, because this is broken down in presence of water, there is energy generated out of electrostatic repulsion between phosphates. Electrostatic repulsion simply means that the phosphates will move apart from each other. Okay. So the potential energy stored in them is converted into kinetic energy. So ultimately we say in simple words that energy is released. And this energy which is released is of course used for plethora of things like cellular signaling. It is also used for formation of phosphodiester bonds during DNA and RNA synthesis. Okay. So this is the reason that why ATP is the energy currency of cell. Okay. Then another question which comes to my mind is what is the role of free ribosomes and attached ribosomes in a eukaryotic cell? Of course, this condition is only possible in eukaryotic cell because we don't find endoplasmic reticulum in a prokaryotic cell to which ribosomes are attached in eukaryotic cell. So free ribosomes will have a role in producing secretory proteins. What do you mean by secretory proteins? Proteins which are to be let out, which are becoming extracellular, which are targeted outside the cell extracellularly. This is what free ribosomes do. And attached ribosomes will produce structural proteins like collagen, keratin, actin, myosin. Because of course these proteins are also chemically modified by adding carbohydrate, by adding fatty acid chain, which is glycosylation, acetylation. There are a couple of chemical modifications. And These proteins also act as enzymes. So ultimately, attached ribosomes will produce structural proteins and enzymatic proteins or enzymes. Okay. This is the functional difference between the two kinds of ribosomes. Okay. Then a question related to this is, usually we write size of ribosome as a value, say ATS of eukaryotic cell. What do we mean by S? So S simply means Swedberg's unit or sedimentation coefficient. Okay. What is this? And how do we measure the value of S? So this is a measure of mass and density of ribosome. So when prokaryotic ribosome is centrifuged, it will separate at a density gradient of 70S. And this is a measure of its mass, density, and somewhat shape as well, or conformation, you can say. While if eukaryotic ribosome is separated by centrifuging, it will separate at a density gradient of ATS. It is going to separate at a density gradient of ATS. So here it means that the mass and density is equivalent to a value of 80. Now we also know that prokaryotic and eukaryotic ribosome would have subunits. So prokaryotic ribosome is made up of 50S and 30S subunits. Eukaryotic ribosome is made up of 60S and 40S subunits. So you might be thinking that prokaryotic ribosome is 70S. So why don't we add up 50 and 30? But 50 and 30, if we add up, it will come up to 80S, which is the value of eukaryotic ribosome. Do we do so? No, we don't add. This is not math. This is biology. So we don't add. When large subunit of any ribosome, prokaryotic or eukaryotic, is separated, it will separate at a specific value of density gradient. Say for example, the large subunit of prokaryotic ribosome is separating at 50S and the large subunit of eukaryotic ribosome is separating at 60S. Same with the values of small subunits of both the ribosomes. So it is separate value of sedimentation through centrifugation of the subunits of prokaryotic and eukaryotic ribosomes. Okay. So we don't add them up. It is simply meaning that the subunits will have their own sedimentation coefficient value. And this unit was of course discovered by Swedberg. So it is also named as Swedberg unit. All right. Then another question which comes to my mind is about what was it? Let me see. We were supposed to discuss about ribosomes. Okay, that's not coming into my mind right now. So anyway, that much for today. I think we have already discussed 3 questions. So if you are interested in such curious content, okay, definitely you can check out the link below and buy my book Curious Corners in Biology, which is available at Amazon. It is a well thought out book, which is a compilation or collection of various questions in this subject of various topics. And so far we are only discussing about cell biology. Okay, there is a whole dungeon of questions remaining to be discussed. So if you find this interesting, please visit the link below. Otherwise, we will see you later. Bye.